Hope everyone had a good break. So we'll continue on here. So the next thing we're going to do is just kind of focus a little more on, how we think about doing these, doing what Dennis talked about from an individual project type of perspective, how we go about planning this. And the goals really are, how do I, oh, okay. It's frozen.
Dennis, how do you?
Keeps clicking.
Oh, there. I think you just clicked the.
Okay.
It's, I don't know.
Okay, so the way we try to approach this, and this is, the whole R24 grant is written around these ideas, is that we want to do proteomics experiments, not just run samples. So, one of the reasons is that, we're all scientists, so it's a lot more interesting to us to think about the experiments than just, open packages of samples and run them through instruments. But we also think it works better if we approach this as a scientific experiment and we talk about how to do it. We plan it out carefully in advance rather than just being a data generating core facility. So we'll start here where this kind of summarizes a lot of what Dennis just talked about. This is, basically what our facility is able to do in terms of, technical capabilities. We can extract protein from lots of different types of samples and digest it with trypsin. We've got a few other options for processing of specific types of samples for various types of experiments. We can do the isotopic labeling and some additional fractionation of the samples. And then we run everything on a mass spectrometer, mostly with the tandem mass tag approach or the data independent acquisition approach. We still actually do use data, label-free DDA quite a bit, or mostly for small pilot type experiments where people aren't necessarily looking for rigorous quantitation. But for, to keep things simple, we'll talk about TMT and DIA mostly for now. And, again, we want to approach this as, a group effort. Everyone is involved in doing a scientific experiment. So there are challenges to that, especially since, we're often working with somebody who's at a different institution in a, different city, different state. So that makes things difficult in some respects, but we think we get an overall better result if, everything is carefully planned up front. So, that's what this slide is emphasizing here. And Dennis mentioned this explicitly. It's much better if we all have a conversation about what the plan is before anything happens and any samples are prepared. Now, that's, one of the things we are accustomed to dealing with on a regular basis is that nothing ever goes according to plan. There's always something that goes wrong, or just for practical reasons can't be done the way you would ideally like it to be done. So if you're thinking, oh no, I've already got some samples prepped in the freezer in the lab back home, we'll still work with you on stuff like that. If you have samples ready, there's often a good result you can still reach. But the reason we want to encourage you to get in touch with us as early in the process as possible is because that's the point at which the maximum number of options is still available. So ideally we want to plan everything out beforehand. So, this will probably work better if we have, an example project to refer back to as we go through this process. So does anyone have an experiment they have in mind that they want to do that we could use as an example? Yes, what?
Okay, so my protein is called CDC42. I have an Ultra ID, which is a biotin labeling tag attached to it, and I have 3xMYC attached to the same protein. So I'm planning to do traditional pull downs and proximity dependent biotinylation for both. But I'm comparing a knockout and a wild type, more or
Okay.
less. Mhm.
Knockout and wild type of the pull down target.
Yes, of the pull
Okay.
down targets. Yeah.
Okay, so pulling down different isoforms of the same protein and looking for how that affects formation of the protein complexes.
No, so I'm looking for the change in proteome. So the protein helps in temperature stress. So when I knock out the protein, the cells are not able to grow at high temperature, and so I'm looking at the change in proteome in those two conditions.
Okay, so you'd have a wild type and a knockout, and then
The complement.
two different temperature conditions. Okay, so two variables for conditions. Then, okay, can we get one more example project? That's a great one, but pull downs are kind of a special case, so we want to have something else too. Yes.
So my project is related to extracellular vesicle. We are extracting the
Okay.
EVs from nasal mucus cell type, and then we will be purifying those EVs using two different methods. The final one will be like magnetic bead extraction. So whatever the samples that we get is through the magnetic bead. So we are not so sure. The first thing is like we don't know how much sample we will get because EV is like very little in quantity, and the cells that we'll be producing will also be like very different too. So that's another one. And the other one is like bacteria-derived extracellular vesicles. So the bacteria, they also produce those EVs, and we also use two different methods to extract those EVs.
Okay.
So we wanted to compare wild-type bacteria extracellular vesicle with another specific strains like MRSA or some other things.
Okay. So at this point, so the experiment you want to do is looking at how the EVs differ from depending on the bacterial strain, but you also need to do some preparatory work to determine, which EV isolation method gives you the best
Better proteomic data.
the best particles and the best proteomics data.
Yeah,
So this
exactly.
could be maybe a two-stage experiment or something like that. Okay, so we can. Yeah, we can work with that. So we've got an IP experiment and an EV experiment. All right. So yeah, that's so basically just, usually if we talk to someone on a Zoom meeting or something, usually, pretty quickly, if you can get to the point where you have a mutual understanding of, okay, what's the biology behind this? What do you want to get out of the experiment? And that's essential to pick the best way to do it. And then, if necessary, we won't do this here because Dennis has just given a lecture on what our facility's capabilities are. But that would be the other side of the interaction is, getting to the point where we understand what you need out of the proteomics and you understand what we can do to get to that point. And then, a lot of this from there on is, similar to how you would plan any scientific experiment. You need to think about, what controls do you need? How many, how many replicates and that sort of thing. So, as Dennis was pointed out in the earlier lecture, kind of our, this varies a lot. How many replicates should you have or can you have? We try to push for at least an N of 5. That's, because, even, 3 may be enough in some cases, but you're always in danger of losing one because either there's an outlier because there was a variation in the prep or something goes wrong at some point. And if you only have 3 and you lose one, then you're in trouble. You can't do all of the statistics and the bioinformatics. But if you have 5 and you lose one, or even 4 and lose one, then you're still okay, at least in terms of being able to complete the analysis. But this is often, we don't, as scientists, we would like to live in a world where budgets don't matter and time doesn't matter, and if you need 5 replicates, you can always prep 5 replicates. but often there are trade-offs in, how much time do you want your student to spend on this, or, what's the budget? It's easy to tell someone, okay, you need to do 5 replicate pull downs for, each of your 4 experimental conditions. That's a lot of work, though, with 4 conditions, it's probably doable, but, there are technical challenges to this. So we often, we'll give you our recommendations, and then, often we have to get to the point where, what's actually practical and what's the best that can be done in a given situation with a particular experiment or sample type. But generally, we want 5. So going back to the pull downs and the EVs, and in both of those cases, pull downs, those are elaborate protocols with many, many steps, and it's very difficult, no matter how technically expert you are at the bench, to do those reproducibly. So replicates in that type of situation, that the sample to sample variability is much higher for pull downs than, say, for 2 million cells in a cell pellet from cell culture. So it actually is, more important in that case, in those cases, to have a lot of replicates. With EVs, it kind of, I don't think we do a fair amount of EV proteomics, and I don't think we've seen the same result twice. There's a lot of different ways to prep EVs, and you're starting out comparing different methods, and there's a purity and quality of sample varies a lot depending exactly how you do it and how much experience you have, how much practice you put into optimizing the protocol in the first place. So those are, with especially with, complex protocols and low level samples, replicates tend to be particularly important. So if anyone has questions at any point, please jump in here. I've learned, I've given enough lectures that I can continue talking for as long as I need to, but it's usually more interesting if we get a discussion going. So please interrupt me at any time. Okay, and then. So then, once you've got an experimental design, you need to, we want to make sure that there aren't any questions that come up later that we forgot to ask about, or, an assumption was made that we can just look for something in the data later without knowing that we were going to need to do that from the beginning. There are lots of potential pitfalls you can get into in that area, but talking about post-translational modifications is a easy way to illustrate that. So if we need to look at phosphoproteomics, to do that properly, we want to be able to enrich for phosphopeptides at some point in the sample preparation and analysis process. So we need to know at the beginning that you're interested in looking at phosphorylation sites or signaling pathways. So it's all this is all it's a recurring theme here. I'm just saying over and over again in different ways that we need to have a mutual understanding of the goals of the experiment and the analysis before we get started. So, kind of a digression on PTMs. So we always, in the field of proteomics always talks about this subject like we're a lot better at it than we really are. Because you can do PTM proteomics, but it's much harder to do in the context that we do proteomics, where we're accepting samples from outside and doing a single experiment, and you have one shot at it basically. To do to comprehensively map phosphorylation sites on a protein of interest, for example, that takes a lot of work. You need a lot of protein. You need to do several rounds of mass spec analysis and try out different proteases, and you can do it. But what we can actually do as a practical matter in a core facility type context tends to be more limited. So we're pretty good at phosphorylation, and it's actually easier to do on a global scale than with a single purified protein. Because the enrichment works pretty well. You don't have to grow up an insane number of cells to get a good yield from that from the affinity purification protocols. And we can look at other PTMs, but as Dennis was saying, we usually push the enrichment step back on the PI because they're a lot more challenging. If you want to look at methylation or acetylation, for example, there are pretty good, enrichment kits out there, but they're antibody based and they require large amounts of input, like milligram levels of starting protein. And so but if you're, yes, question.
Oh, sorry, I have a simple question back then. So when you say replicates, do you mean that like replicates within the same sample or different samples?
Okay, yeah, so yeah, I'm glad you asked that. So when we say replicates, what we mean is biological replicates. And when we say sample, we just mean whatever's in a single tube. So so if you're a biologist, when you say sample, that probably means an experimental condition, and then you have X number of replicates of that sample. So we use the terms a little more
Okay.
loosely than.
So for us,
that.
it's number of subjects.
Yes.
thank you.
So for a cell culture experiment, biological replicate would be independent cultures. For pull downs, it would be, 5 different pull downs of the same experimental condition rather than one big pull down and splitting it into 5 technical replicates. For the EVs, it would be, 5 independent preps. Okay. So yeah, okay, I've probably talked enough about PTMs, but the take home here is, for if you're interested in PTMs or anything other than looking at protein levels globally, we want to talk about that in advance. So then at some point we need to figure out, so we've got these different options for how we do the sample preparation and the mass spec analysis. So which one is the best one? So DDA, data dependent acquisition, again, we use that sometimes. That's, typically for a preliminary type or pilot analysis where you're not going to do a full blown bioinformatics workup. But the 2 main quantitative options are TMT with the isotopic labeling, or the DIA where you do the M over Z window acquisition, skipping past the initial MS peptide detection. And so on this slide, in previous years, the TMT and the DIA in the title have been the same size, but I made the DIA much bigger this year because the answer to the question, which one of these should you use, is increasingly DIA. I mean, the advantages of TMT are just not as great as they used to be for a couple of reasons. One is just, mass spectrometers have gotten much faster and much more sensitive. So the advantages of that isotopic label just, are overcome by what really fast scanning in DIA mode is able to accomplish. But from the perspective of planning out an experiment, you definitely want to you want to have this discussion. You want to reach agreement on, okay, this is what we're going to do for this experiment. So going back to the pull down example, it's possible to do TMT with pull downs. Usually, you label the input and then pull down the labeled samples. We don't do that. We've just never developed the protocol because it's complicated and difficult. It would mean either you have to do the TMT labeling, which most non-mass spec labs don't have any experience with, or we would have to do pull downs after labeling here, which we don't know how to do very well. So most low-level samples, we lean toward DIA because it works better for the detection is better, detecting low level peptides potentially underneath higher background, more abundant protein peptides that, DIA is better than at that. And we don't have to worry about, whether we have enough sample for, efficient isotopic labeling in the first place. So most pull down experiments we'll do by DIA if we have sufficient replicates. The EVs, basically the same thing. It's going to be, depending on your source, maybe you can, I don't actually know how many, you can probably get a pretty good sized EV prep from bacterial cultures, I would guess. I haven't, so that may not be as much of an issue. But, you're usually sample limited with EVs, so we're going to, go for the DIA option there. So what for TMT, at least for now, we're still doing phosphoproteomics that way, though that probably won't be true for much longer. And then, TMT is also useful if if you have, for a small experiment where you want, you can, if you have, a wild type and a knockout and 5 of each, that's a good TMT10 experiment. You may, you'll still get a really nice quantitation with, problems in the data analysis with missing values tend to be minimized by TMT. So it's, it's still useful in some cases, but increasingly as we see larger and larger experiments with more and more samples, it's we're using it less and less. Good. All right. So, once we get to, yes.
Can you turn the microphone on? Sorry.
I just have a quick question about TMT labeling efficiency. What is that like?
Oh, it's the chemical reaction is very efficient. If you have enough sample, then you get 90+% labeling at least, even.
Yeah, I would say it's probably, between 95 and 99% efficient. And I also say it's so easy to do that even I can do it. So if I can do a chemical reaction in the lab, it's really easy. But the one of the problems is you have to know how much protein you have and you have to control the label. And so there can be inefficiencies if you have more protein than you thought and you didn't use enough label, it'll be under labeled. And then the opposite. So if you don't have as much protein as you thought and you use too much label, you can actually have over labeling. So instead of only hitting primary amines, it'll also, you'll get a reaction to some side chains.
Okay.
And so keeping that ratio optimal, which Casey's not in here, right? So it's not the protocol that Thermo sells you. So that's not optimal.
Okay.
Actually, about a fourth of that is optimal. So you can label 4 times more protein than what the kit says. So we actually split kits across multiple experiments. And that's one way that we're able to keep the cost low. It's still expensive, but we're still stretching the kits more than what their Thermo intends.
Okay. Thank you.
Okay. So then, so once we've got an experimental design in mind and a basic workflow planned, so one thing, one additional thing we want to spend a lot of time and care on is making sure that we have good samples. So there's the problem, a lot of what we do in proteomics, the quality of the data you get in the end is determined by the quality of the samples you start with more than any other factor. So the fact, and there are a lot of dimensions to this, and we're not just talking about did your first year grad student know how to pipette well enough to do the protocol right, but what we really mean more than that is just were the samples prepared, collected, et cetera, in the right way to be optimally compatible with the processing we need to do and for collection of mass spec data. That's really mostly what we're talking about. I won't get too much into the weeds on this because Karen's going to talk about this in greater detail in her talk. But basically, for any different sample type, and there, this is not exhaustive here. We can do a lot. There are other sample types we can work with that aren't on here, like EVs. But for any specific sample type we accept, we have protocols that we've already optimized and had in place usually for a long time that we know work well with those sample types. If those samples are prepared in a certain way within certain mass ranges or cell number ranges or whatever it may be. And this is changing over time because one of our goals is to be able, we want to be able to work with less sample than we can now. Proteomics is pushing toward, single cell, just like genomics has. There's kind of a debate, or this is something else Rick can talk about at length, is, how much are people really going to want to do actual single cell proteomics? But we know we want to be able to do 1,000-cell proteomics or probably even 100-cell proteomics. Right now, that's hard to do. It can be done, but doing it reproducibly, reliably in the setting where, in a context where you're doing, you're having people send in samples and we're doing this routinely, it's still a challenge at this point. But we expect in the next, couple of years or so, we'll make a lot of progress toward lower level samples. And we've purchased equipment and made plans to develop those lower level protocols. So that's kind of where we're going. So this will change over time. But just to illustrate,
Sir, I have a question.
yes.
Yes. So what is the minimum amount of protein that we should start with for the experiment, for the proteomics experiment, considering the sample loss at various stages? And what is the minimum amount of peptides that we put into the mass spec? For the, if we want to run 3 replicates, technical replicates, what is the minimum amount of?
Very important question. And so that sounds simple when somebody asks, okay, how much protein do you need? There's how much we need and how much we want. So, the simple answer that we'll probably give you if you ask how much protein we need is 100 micrograms. Because that gives us plenty to work with. It's actually several times what we need, but we always want more. 100 micrograms is plenty. We can, that gives us enough for TMT if we want to do it. It's easy to do DIA.
That can also include the phosphoproteomics.
Yeah, for phospho, at least the TMT based version, we want more. So that's for phospho, we'll ask for. We want to have at least 200 micrograms so we can get a good phosphopeptide enrichment. Hopefully those sample requirements will go down as we transition to a label-free version, but we're not quite sure yet what the input will need to be.
And what is the amount of peptide that you put into the LCMS?
Right. So our LCMS column capacity is about 1 microgram. So most of the sample requirements, when we say we need 100 micrograms, most of that is just because that's how much we need to get the sample processing to work well and still give us enough left at the end after several steps of processing that we have more than enough to inject on the mass spectrometer. So that this is actually the real obstacle to single cell proteomics. It's not that the mass spec isn't sensitive enough. They have been for years now. The problem is how do you prep the sample, a single cell, extract the protein, and get it into the mass spectrometer without losing it. So we actually don't need very much protein once you're able to get it into once it's in the mass spectrometer.
So 1 microgram is the 1 microgram of peptide is what we inject into the LCMS.
Yeah,
for the 3 replicates.
That's the maximum we can we can work with a lot less. 100 nanograms injection, you'll get fantastic data out of that. 10 nanograms, you'll still get pretty good data. And depending on what the objectives of the experiment are, you can often work with much less than we say we need. So kind of guidelines. So 100 micrograms, that's ideal. 30 to 50 micrograms is still really plenty of protein for most applications, except maybe phosphoproteomics. 10 micrograms, that's still pretty good. We can, fairly routinely get good data out of 10 micrograms a sample.
10 microgram of peptide digest you are talking about? 10 microgram?
No, I'm talking about input sample, like the amount 10 micrograms of total protein per sample that we start with at the beginning of sample processing.
Okay.
I was going to say that one of the main limitations there is our ability to do a good protein quant. And so that's, if you gave us 20 micrograms of protein, we're probably going to use half of it to get a good protein quant, because that's how we normalize the samples from one to another. And so that step is crucial. And so that's really kind of where we're limited when it's down at the 10 microgram level. We're just going to digest everything and then try to normalize the best we can on the mass spectrometer response. We're going to assume that it's the same input, but that's really where some of the, and if we do that, there's going to be more variance in the data, and so it's going to be, the outcome's not going to be as good. So the more we have, the better the results are going to be.
Thank you.
So on the lower end, those, and this is a challenge with pull down samples, is you often have a very small amount of the protein, or at least the protein you're interested in. Often you can't do a concentration measurement after the pull down step, because your proteins are still on the beads, or even if you can elute, there's not enough there for a concentration measurement, or if there is, it's just the non-specific background that you're actually measuring, which doesn't give you much useful information. So you can't, I'm probably going to get in trouble for saying this, but you can do a proteomics experiment with, you have a very low amount of protein. You don't really know how much. You have one shot at it, and we'll just process the samples, run them, see what happens. Sometimes that works, sometimes it doesn't. So you don't want to do that unless there's no other choice. And so with pull downs, you're kind of limited in how you can characterize the samples up front. But anytime you can, you want to prepare more protein than we actually need, and do a, carefully quantify it and characterize its, quality, purity, et cetera, where that's possible. Okay, so just to, illustrate the problem of, sample contamination. And again, here that this just means stuff we don't want to have in the sample. So on the left, this is just, to illustrate, if you've ever run an SDS page gel, the top panel, this is what it's supposed to look like. But if you have, a bunch of, extra detergent or salt or whatever in there, it can come out looking like this bottom panel, which is, definitely not what you want. It messes up your resolution and your, how well the gel runs, basically. And the same principle applies to LCMS. So this is the top panel here. This is similar to what Dennis showed you in the last talk. That's what a 1 hour acquisition is supposed to look like, with peptides well distributed over the hydrophilic hydrophobic gradient. But if you've got something you don't want in your sample, you can end up with something like this. This is what happens if you inject detergent into across a C18 column, and you just you don't see what you need to be able to see. So we've got.
Oop, I've got a couple questions on here too, so I'll let you. Do you want to finish your thought, or?
No, let's
Okay,
go
we're
ahead.
moving on. So Mike mentioned that you might want to mention the difference between quantitative experiments and much simpler qualitative identification experiment.
Yeah. So we'll, we may not get through all the slides here, but that's fine.
There's one more question.
Yeah,
too.
I was just talking about the context between the amount of proteins that you need. That Rick mentioned protein quant and the fact that that's somewhat wasteful, not wasteful, important, but uses a lot of protein. And that's just a requirement of doing good quantitative analysis. In the old days of just doing quantitative analysis, we often didn't even know how much protein we were getting. We just took what we got and ran and identified as many things in it as possible. And so I don't know if some of the post-translational people are thinking in terms of that it's mapping, at this stage, are they just mapping their phosphorylation sites, or do they really want to set up to map and then do a quantitative comparison between a couple of different conditions?
Yeah. So, in these workshops, we talk a lot about bioinformatics and quantitative comparisons because, that's what we do. It's, that's where a lot of the time and effort and expertise comes in is, what do you do with the data after you've generated it? But, there are a lot of situations where you just want some preliminary data, something you can, put in your grant application that shows this is possible to do, or you're in a, it's a preliminary experiment and you just, with a, like a pull down experiment, for example, you may want to do that quantitatively, or you may just want to see, okay, I just want to keep this simple, do a quick experiment, get a list of proteins that are present in the, control versus the experimental pull down. And that, the considerations there for how much sample you need, what we're going to do, how we prep it, how we acquire data, those can be very different. So there are there's a range of options available for, how to economically and effectively generate, preliminary, just protein ID data versus a rigorous quantitative experiment. Yeah. So what was the other question?
If I want to examine changes of different isoforms for the protein I'm interested in, can current proteomic core do it?
That's challenging, looking at comparing different isoforms of the same protein, because, basically, if you have closely, very, very similar isoforms, most of the tryptic peptides generated from those two different isoforms are going to be identical, and we won't be able to tell which is which. So you'd have to do that in a targeted way. You'd have to, verify that you can detect, reliably detect, one of the peptides that is actually different in sequence between the isoforms. And then, we'd probably kick that over to Mike to do a targeted experiment on. And, it's doable. Yeah, it's proteomics can do that, but you wouldn't, in a typical discovery experiment, looking across, 6, 8, 10,000 proteins, you're not going to be able to tell which of two closely related isoforms is present in the data most of the time. Okay, so we'll, I'm running out. We're not, okay, well, I won't unnecessarily run over time, but I'll.
Karen's next, and she's like, no, it's all the time.
The critical question at this point, I think, is lunch here yet or not, but that's what people are really going to want to know. Okay, so, I will skip over this slide because I can, cover that concept in the instrument time slide. But so wrapping up this, this question of how much sample, that's a moving target. So, 3 years ago, we would, want 1 million cells, 100 milligrams of tissue, 50 microliters of serum. 3 years later, we can, we still might ask for those numbers, but our ability to handle much, orders of magnitude less sample is significantly better than it was 3 years ago. 3 years from now, we expect to be again much improved in our ability to handle just a few cells or very small tissue or serum samples. The IPI, it's all question marks because you never know how much protein you have. And then, we can gloss over this because we kind of covered this concept. This is really emphasizing that, you need to, ideally you want to start with a lot of protein so you can have options. You can, you know how much you have, you can process it well, you have room for error with sample loss, or you can, so Rick likes to quote his rule of halves, or is that what it is, where you want to be, you want to assume you're going to either lose half your sample at each step, or you're going to mess it up and have to repeat it. So you want to start with several times more sample than you really need. And then what we actually analyze, it's usually just on the microgram level, or sometimes much lower on the mass spectrometer. And then, all of this comes down to, so we have a lot of flexibility in, which instrument we use, how much instrument time. We can fractionate samples and run fractions. So we can put 24 hours of instrument time into a single analysis, or just, 1 hour per sample. Right. Yeah. The better the instruments get, the less instrument time you need to generate equivalent data. So we're able to design a workflow, pick whatever, we can tune whatever experimental parameters we want to fit a budget, or to fit experimental design needs, experimental objective needs. So that, the basic challenge is, we can identify 10,000 proteins in a lot of different sample types, but we don't really want to do that unless we have to, because it's expensive. It's getting less expensive. But that's challenging, because you usually don't know how many proteins you need to identify until after you've done the experiment. But sometimes you can make a pretty good guess, and then we can come up with, for any different experiment, it's potentially immensely flexible in exactly what we do. And that can, so whatever your needs are, whatever you want to get out of the experiment, however much you have to, in terms of time and budget resources to throw at it, we can probably find something that fits that situation well. And then, I think this is the last, or the second to last slide. So, current capabilities, we've got, 3 different workhorse instruments. We've still got a couple of older ones, but we'll just focus on these. So what can we get with TMT? We usually run that on the Eclipse instrument, typically 6,000 to 8,000 proteins from, mammalian cell lysate global analysis. DIA, until just recently, we did mostly on our Exploris 480, and that got us into usually the 4,000 to 6,000 protein range. So that was why, TMT was really useful when you wanted those extra, couple of thousand protein IDs. But we've just recently installed Thermo's latest and greatest Astral, which can get you, up into the 8,000 to 10,000 range in 30 or 40 minutes label free. So this is why, TMT is much less useful than it used to be. So really, I mean, once we've made the investment of buying the instrument, we can get more protein IDs than we could just a year ago, for really the same kind of budget that we could, it doesn't take it's not more expensive now to generate 10,000 proteins than it was a year ago to get 6,000. So this is moving pretty fast, and, a couple of years from now, we expect to be, I don't know how far you go. Maybe, it's probably hard to get past about 12,000 protein IDs, because they just aren't there. But, we'll focus on, other things that we start to be able to see, to be able to do better when we have this capability. So yeah, question.
My question is about label-free quantification. So especially for the IP samples, or, PTMs, where we don't know it, looks like you are digesting it and trying to find out the protein quantification, and that's what that is. But many of the other publications, they go for SciLac if they really wanted to do so I just wanted to know from you, like, how much trust we can have in this label-free quantification versus, SciLac.
So the label-free quantitation is based on peptide peak intensity. So that's, that, So if your chromatography is good at the, in the LCMS run, you can, we've seen some of these figures where you can see the actual peptide peak. So you can imagine you could, just measure area under the curve for each of those peaks. That's basically, it's much more sophisticated than that, because in a cell lysate, you have, thousands of overlapping peaks of different intensities over several orders of magnitude range. So you need sophisticated peak picking software basically to sort that out. But it is doable, and the sophistication, the quality of that analysis has improved over the years. So for a long time, this DIA technique has been around for a long time, but it's been, in typical biological type experiments like the context with the area we operate in, it's been of relatively limited use. TMT has always been better until fairly recently, but as the scan speed of mass spectrometers has improved, so you can acquire more data in less time, and the sophistication of the data analysis software has improved, especially with machine learning techniques entering into it. It's really good now. So this is one reason we want a lot of replicates, to ensure that, so we can see that there's, replicates clustered together. We don't have a lot of variability coming into the analysis, but it is quite reliable, and our bioinformatics people or Rick can get into the exact details of that as much as you want. But it's if it's done right, you have good chromatography, you ensure that you're scanning enough points across your peaks, you can define those peaks well, and you can quantify them very, very reliably. Okay, yes.
My question is, what method to recommend to do the phosphorylation enrichment? For example,
For phospholipid enrichment, so we do that after trypsin digestion. So we enrich for tryptic peptides that are phosphorylated. We use the thermo high select. We actually do it twice. We use the iron based, and then what's the other one? The titanium oxide based.
which one works better in your hand? Both.
We use titanium first, and then whatever comes through, we use the iron, and then we combine them.
So
Yeah.
if we send you pallet, that stage is done at your place, right?
Yeah.
Yeah. We won't do the enrichment for any other PTM, but for phosphorylation, we do. So I mean, the two methods are, I wouldn't say one works better than the other, but you get slightly different enrichment. They're not, there's not perfect overlap between the two. So we do both and then recombine.
Yeah, one of them is more for multi phosphorylated peptides, and the other one is for single. So it pulls down all of them with a combination, or most of them.
But I guess it's, this is a little off topic, I guess, but phosphoproteomics is not completely unbiased. So since we're digesting with trypsin, we will only see sites that are well positioned on good tryptic peptides, and the enrichment methods they work better for some peptide sequences, some than others. So it's there. You can usually get, we can identify, tens of 10,000 plus phosphorylation sites in a typical analysis. So if you're interested in a specific signaling pathway, you'll usually see enough of it to be useful. You can get good data out of it, but it doesn't work perfect. It doesn't comprehensively identify every phosphorylation event in the cell.
Yeah, and we have a talk tomorrow that we'll go into more detail about that.
Okay, so yeah, this is definitely the last slide. So, this is, we won't spend much time on this, but one of the challenges is, so once we've done a proteomics experiment, we've generated data, there's usually a follow-up meeting, because the initial reaction to receiving one of these data sets is, okay, what now? Because it's an enormous amount of information, and you can start analyzing it and keep analyzing it for as long as you want to, basically, if it's a good data set. So there need to be, understandings and parameters and limits on, how that's going to be done. And, there should be, goals and objectives there as well. So you don't get into endless loops of coming back and reanalyzing old data sets because, there's limited capacity to do that. So you want to get to the point where you've answered your original question to the, as best you can, or you've got what you need for your grant or to round out your publication. Okay, so we'll stop there and